Dear students, welcome to Dr. JPL video lecture series. This is the last video related with Southern blotting from Unit 4A Immunoblotting Techniques as per Pharmacy Council of India syllabus for third year B Pharmacy, 6th semester, subject Pharmaceutical Biotechnology and subject code is BP605T. Dear students, myself Dr. J.P. Lavande, Associate Professor, Department of Pharmaceutics, Shivlingeshwar College of Pharmacy, Almala, Taluka Ausa, District, Latur. Already we had covered the details associated with the blotting techniques, the general procedure for blotting, what is southern blotting, what is its principle involved, and in this video we will go through the detailed procedure for southern blotting and its applications. Once more we will go through the principle in case of southern blotting. So restriction endonucleases, which is an enzyme, is used to break the DNA into small fragments. These fragments are then separated using electrophoresis. The fragments achieved is then classified according to their size in the form of kilodaltons. Thus, DNA fragments are transferred to the blotting paper where it is incubated with probes. Probes used in the southern blotting can be highly selective. They can selectively bind with a resolution of 1 in million and the characteristics to bind to the intended target fragments. So the different steps involved in southern blotting procedure are step 1 to step 6, which follows through the DNA purification, fragmentation, gel electrophoresis, denaturation, blotting and the hybridization. The DNA purification, that is the generation or isolation of the DNA from the tissue or cell. The fragmentation will consist of applying the specific restriction endonuclease enzymes onto the DNA so that the long DNA that can be fragmented into the small stretches of DNA. Then these are subjected through running in the process which is known as the electrophoresis, and they can be separated as per their molecular weight. Then the fragments will be subjected to the process of denaturation, that is converting from double-stranded DNA into the single-stranded DNA form, followed by blotting onto the nitrocellulose or nylon filter paper, followed by the hybridization with that of the radio-labeled probes with the phosphorus 32, and then subjecting it to the process of autoradiography. So this is the flow chart how the blotting process that can be carried out in the form of different steps from step 1 to step 6. So step 1 that will consist of, it involved to extract the DNA present inside the nucleus of a cell. We must first lyse the entire cell to enable the expulsion of the DNA. Incubating the cell culture with detergent lyses the entire cell. Now the lysed sample contains DNA, proteins and debris. Protein is lysed by the addition of proteinase enzyme and incubated. DNA is purified and separated by alcohol precipitation, and fibers are removed by using a buffer. Apart from standard manual isolation procedures, there are commercially available kits like Gel Electrophoresis for the isolation of DNA from a variety of sources such as mammalian cells, plants, bacteria, fungi. High purity of the isolated DNA is ensured in case of use of commercial kits. The step 2 consists of fragmentation. The long nucleotide sequences should be broken into smaller fragments for the purification or identification process. This is done by the restriction endonuclease enzyme. All the reagent necessary for the digestion process should be kept on ice before setting up a restriction digestion reaction with suitable enzyme and appropriate DNA concentration. The components are added to the multi-well plates or microcentrifuge tubes, that is PCR tubes, and mixed by aspirating the contents with the help of a pipette slowly to avoid formation of any bubbles. It is to be taken care that the enzyme has to be added at the last step, and until then it should be stored at -20 degrees Celsius. To ensure complete digestion of the DNA, a surplus amount of enzyme is supposed to be added, as the fragments produced due to partial digestion can cause ambiguity in the results, leading to inaccurate analysis of the subsequent blot to be analyzed. Nevertheless, the concentration of the enzyme added in total should not surpass one-tenth of the total volume of the digest, because the glycerol content generally found in stock of the enzymes has the capacity to impede digestion process when present at high concentration. Also, in order to minimize the probable errors due to pipetting, master mixes should be prepared accordingly when there is a requirement of analysis of a large number of samples. The incubation of digest is carried out at 37 degrees Celsius either in an incubator or a water bath. A water bath is generally preferred for DNA samples obtained from cloning 1 to 2 hours deemed sufficient. In case of genomic DNA, there is a requirement of overnight digestion, wherein there is a chance that the enzyme is over in between. To ascertain that the overnight reaction is successful, half of the enzyme is to be added at the beginning of the digestion reaction, and the second half can be added in the morning, and the incubation can be continued for an hour. Once the digestion is over, it may be the case where a concentration step is required to ensure that suitable volume of DNA is present for loading into the gel, which is generally fixed at 20 microliter per well. This makes the total volume to be in 24 microliter after the addition of 6-time loading buffer. A standard method for concentrating the DNA samples involves precipitation in presence of ethanol, after which the DNA sample is resuspended in water. The traces of ethanol should be removed completely, which otherwise would lead to spilling of the samples out of well once the samples are loaded. Step 3. Gel Electrophoresis. Nucleic acids are negatively charged molecules, so they move towards the anode in an electrophoresis chamber. The movement of the DNA fragments differentiates the rate of the transport, thus enabling the separation by size. The percentage of the gel that is to be used and size of the gel has to be determined. The percentage will decide on the size of the fragments that will be separated, and the size of the gel, on the other hand, will give the range of fragments that are feasible to be resolved. Longer gels are generally needed in the case of separation of genomic DNA or multiple fragments that are having similar sizes in order to guarantee appropriate separation. Generally, a 0.7 to 2% gel is considered to be adequate for most of the applications. However, in case of some genomic DNA samples, it may be required that a low percentage of gel is supposed to be run for the necessary separation of the fragments. When a less than 0.8% gel is required to be run, a high base gel percentage is required, up to 2%, to provide support as low percentage gels are very fragile. It is necessary that the base gel is dispensed before the comb is positioned. As soon as the base gel is set, the low percentage gel is poured on top and the comb is placed in a way that it does not touch the base gel at the bottom. Step 4. Denaturation. DNA thus attained are double-stranded in nature. For our purpose of probe hybridization, we need a single-stranded DNA. DNA is therefore denatured in an alkaline solution. This results in the formation of denatured DNA, that is single-stranded DNA. Step 5. Blotting. Blotting is the transfer of the fragmented DNA sequences to the nitrocellulose membrane or nylon membrane. The process is done by either electroblotting or capillary blotting. The DNA molecule is saturated using a sodium chloride solution and permanently fixed using either UV radiation or drying. Choice of the membrane for blotting. In the original protocol, nitrocellulose membrane have been used for blotting in case of southern blot, but in recent times, nylon membranes have been implemented for the blotting process due to their ability to bind more amount of DNA efficiently, which allows the southern blot to be carried out with less amount of target DNA. Step 6. Hybridization. Labeled probe is added to the membrane buffer and incubated for as it takes several hours for the probe to find the exact target sequence. The time required for hybridization usually 1 to 16 hours depending on factors like complexity of the probe and concentration. The radioactive RNAs are present in limited amount as it is derived from cells, making it an important factor for the original protocol. Due to this, the volume of the hybridization solution used is supposed to be less so that the desired RNA concentration can be achieved. There can be two approaches for hybridization experiment where one uses smaller volume and the other utilizes a vessel destined to hold the membrane in low volume of liquid. The suitable setup possesses a plastic bag that can be sealed using heat, and it consists of optimum amount of hybridization mix and can be dipped in a water bath or a hybridization tube with capped ends. In case of the tube, it must be ensured that the membrane is wet when the tube is subjected to rotation cycles in a rotavir oven. The probes applied can be either RNA or DNA. The cells act as a source for the RNA, or alternatively they can also be obtained from clones or PCR products via in vitro transcription. In case of DNA, they can be either clones or PCR products. The procedure adopted for the labeling method relies on the other source from which the probe is derived. Southern blotting applications. Southern blotting is used in a number of applications. The primary usage of southern blotting is to identify a specific DNA in a DNA sample. It is mostly useful in the identification of viral infection and certain bacterial infections. In rDNA technology, the southern blotting technique is used to isolate a particular DNA. It is also useful in the study of mutation and gene rearrangement. This property is used to diagnose neonatal diseases and genetic diseases. Due to the precision in DNA identification, this technique is used in phylogenetic studies, paternity and maternity analysis, forensic studies, and personal identification. Southern blotting can be applied in studying structure of a gene or to elucidate restriction enzyme maps. Particularly, Southern blotting can be used in the identification of the methylated sites present in case of some genes in particular. Applying restriction nucleases such as MSP1 and HPA2, which can both identify and cleave among the identical sequence, this can be implemented. The discovery of RFLP, that is restriction fragment length polymorphisms, by Southern blotting has helped in the mapping of several genomes that were crucial to be mapped. In the arena of immunology, the clonal rearrangements of the immunoglobulins as well as the T-cell receptor genes that play an important role in elucidating an immune response can be analyzed by Southern blotting. So dear students, this is the last lecture related with the series of Southern blotting, and in this lecture we saw the detailed procedure for Southern blotting and applications of Southern blotting. Happy learning. So dear students, thank you for watching. If you like this video, please hit the like button, share our videos to your friends and subscribe to our YouTube channel Dr. JPL Video Lectures by pressing the red colored subscribe button to get notification about more such videos. Thank you, stay safe, take care and happy e-learning.